Our next speaker is Tim Anderson, who is going to also be talking about malaria.
Good morning. Thank you very much for the invitation to come and speak, and also to Callie for introducing the organism. I'm up here as a spokesman. Other people have done most of the work I'm going to talk about. They are listed down at the bottom of the slide. So it's my lab, TBRI, in San Antonio. I have collaborators in Southeast Asia, in Cambodia, in Thailand, and in Laos. And I work in quite close collaboration with Mike Ferdig's lab in Notre Dame. So malaria control is really sort of riding a wave of success at the moment. So incidence of malaria has been dramatically lowered in many countries in the world. There has been injections of money and enthusiasm from the Bill and Melinda Gates Fund, and that has really goaded funding agencies and governments to follow suit. So things are looking good in very many ways, but there is a cloud on the horizon. The successes that have been made have been largely on the backs of the introduction of artemisinin-based combination therapy. Now there are hints that there is emerging resistance in Southeast Asia, and which is putting a little bit of a damper on the euphoria. So what I want to do in this talk is tell you something about the epidemiology and genetics of artemisinin resistance, and tell you about the approaches we are taking to try and identify the genes involved. So first, a quick Malaria 101 slide to sort of build on what Callie talked about. I think an important thing to emphasize here is that these are obligately sexual eukaryotes. So they have good, upstanding eukaryotic sex rather than the sort of sleazy cut-and-paste bacterial sex that I think we're going to hear about. There's a high recombination rate. So one centimorgan is equal to about 17 kilobases. That's about 50 times the recombination rate in humans. So there's a lot of reshuffling of the genome. There are large numbers of parasites in infected people. So you can have up to 10 to the 12 parasites within a person. So enormous capacity for adaptation. The generation time from sexual phase in one mosquito to sexual phase in another mosquito is variable, but generally around 6 to 12 weeks. And we have laboratory culture, and we can manipulate parasites in the lab. So why is everyone interested in artemisinin resistance? And I hope by showing this slide, which shows the depressing decline in efficacy in Southeast Asia, I hope that'll show you why we are worried about artemisinin resistance. So this shows cure rates on the Thai-Burma border. So chloroquine was introduced after the Second World War. By 1980, that had become useless. The next drug up was sulfadoxine pyrimethamine that Callie talked about. That was introduced in the 70s, and in just 40 plasmodium generations, the efficacy had dropped to zero. Quinine has been used for a few hundred years. Even that has dropped in efficacy on the Thai-Burma border. You may know of mefloquine as a drug that causes frank psychotic episodes in about 2% of patients. It's a very useful drug, and it's been widely used in Southeast Asia. That has declined in efficacy. And the only thing that has really restored therapeutic success is the introduction of combinations using artemisinin. And you'll note that the combination that is still used in many parts of Southeast Asia is with mefloquine, which is a drug that we know has impaired efficacy. And that just shows you how few drugs we have available. So really everything hangs on the efficacy of artemisinin combinations. So what is artemisinin? So it's a plant-derived compound. It was, in fact, the person who really developed it won a Lasker Prize in the past month. It's an extraordinarily potent antimalarial. In 48 hours, it can reduce burdens of malaria by a factor of 10 to the 8. So when susceptible parasites are treated, 95% of patients treated will be clear of parasites. They will be slide negative in 2 days. But a few years back, there were reports of delayed clearance. There were reports of patients who are still infected and slide positive after a week of treatment. And those reports were confirmed in a series of very carefully conducted drug efficacy trials. There really is very, very slow clearance. And in fact, the majority of patients in Western Cambodia show very slow clearance. And that is associated with a much higher treatment failure rate. So it's genuinely something to worry about. And WHO and the Gates Foundation, they have come together and they've set up really a military-style operation to try and build a fence around this focus. And essentially, they intend to try to eliminate malaria in this focus to remove the problem of resistance. And the reason they're so worried, resistance to chloroquine was originally first seen on the Thai-Cambodia border. Same with sulfadoxine pyrimethamine, same with mefloquine. In the case of chloroquine and sulfadoxine pyrimethamine, it spread across Southeast Asia and then overspilled into sub-Saharan Africa, completely changing the landscape of malaria control. So there's legitimate worries. So I want to tell you something about the phenotype, and it's a bit of a strange phenotype. So I told you it's associated with delayed clearance. So to illustrate that, here, this graph shows data from 2 patients. There's a fast-clearing patient, shown by the red dots, and a slow-clearing patient, shown with the black dots. And this is real data. And this is collected by essentially taking blood samples from patients every 6 hours following treatment and measuring parasite density. So we can measure this clearance rate simply by plotting it on a log scale and measuring the slope of the decay in parasitemia. We can also measure the infection half-life, and that's log 2 divided by the clearance rate, which is perhaps a more easily understandable way of looking at it. Interestingly, with most bacteria and all other antimalarials, you can take parasites into the lab, you can grow them in 96-well plates, you can measure the amount of drug that reduces growth by half. With artemisinin, that shows absolutely no correspondence with clearance rate within patients. It's not a biologically useful measure. So we have to do this incredibly laborious measurement. So first question we wanted to ask. We're trying to eliminate malaria in Western Cambodia, but has resistance already emerged in other parts of Southeast Asia? So I work mainly on the Thai-Burma border. That's 1,000 miles to the west. And we work with a group whose job it is to optimize treatment therapies for malaria in the face of multi-drug resistance. And they have been laboriously measuring clearance rates at 6-hour intervals in 3,000 patients for the past 10 years. And I plotted the half-lives against time. And you can see a great cloud of points. But what I hope you will see is in the past 3 years, there are many more patients that show very long clearance half-lives. And for reference, on the right-hand side, I show data from Western Cambodia. Many of those patients in Western Thailand show actually much slower clearance rates than you see in Western Cambodia. And the boxes there simply show patients for which we have very good sampling of blood, so we can actually do some genetics. So I plotted that out just as the proportion of parasites above a threshold. And again, you can see there is an accelerating rate of, well, there are more and more parasites showing very, very slow clearance on the Thai-Burma border. So it's a little bit complicated when you have to do everything in patients because we're treating with combination therapy. So one possibility is that the change we see is caused by changes in response to the partner drug and not actually to artemisinin. Well, fortunately, most of these patients are actually treated with monotherapy, with artemisinin monotherapy for 2 days. So we can measure clearance during that 2-day window, and we can see those white bars clearly show that it's response to artemisinin that apparently is changing. So to cut things short, resistance or this clearance phenotype is emerging elsewhere in Southeast Asia. But many things can potentially affect clearance rate of parasites. So drug concentrations, immunity. We were particularly interested in immunity, in that levels of transmission have been driven way, way down over the past 10 years. So one possible explanation for this is that, in fact, people no longer have immunity to help patients, to help expel parasites. And so this delayed clearance phenomena might even be just merely an artifact of a low transmission and lessened immunity. So we wanted to really see, is this a parasite genetic trait or is it caused by something else? So we've taken a sort of classical genetics point of view, and we've essentially done a twin study, except for in this case, we're not interested in human genetics, we're interested in doing a twin study with genetically identical parasites infecting different patients. In Southeast Asia, even though there's obligate sexual reproduction, there's actually very low transmission and there's a lot of selfing. So we have identical parasites mating together, and that obviously causes no change. And so you find many patients who will be infected by exactly the same parasite genotype. And we can look at them and see whether, in fact, genetically identical parasites tend to show the same clearance phenotype. So we took 1,700 parasites, blood spots, and we genotyped parasites using 96 markers with Golden Gate. We have 93 robust SNPs, all are variable. And we've simply considered parasites to be identical by descent if all 93 of those are identical. And the tree is simply based on shared alleles. And it shows the 158 parasite genotypes that we found in one or more patient. And in fact, we found them in between 2 and 14 different patients. So we have really quite a big identical twin or identical triplet parasite study. You'll see with the genotype data, I've just shown one particular SNP marker. We see clusters of alleles at A and T. We also see a cloud across the middle. Those are cases where we have multiple clone infections, and we've actually excluded them from the analysis. So, do genetically identical parasites show similar clearance phenotypes? The answer is yes, very clearly. So the graph on the top plots clearance half-life. Then I've simply ordered the parasite genotypes along the x-axis. And so you'll see there are multiple patients infected with the same parasite, and they tend to have similar clearance rates. We've got some spots in black. Those are in the last 3 years. We've got spots in blue. They are from 2001 to 2004. And I'd like you to see we've got very slow clearing parasites on the right-hand side that I've marked with arrows. They are genetically identical. They have the slowest clearance of all the parasites from the early period. So it looks like genetics, alleles for slow clearance were already present about 8 years ago in this population. And we can essentially do an analysis of variance. We can estimate then the proportion of variation in phenotype that is due to parasite genetics. And when we do that, we see, that's shown on the bottom graph, we see that in 2001 to 2004, there is very little heritability. It's around just about 0.3, but it's barely significant. When we go to the last 3 years, we see 66% of the variation is due to parasite genetics. This really is a hardwired genetic trait, and therefore it's something that we can map and locate the genetic underpinnings. So the conclusion here is really that we have an excellent parasite population now if we want to go forward and try and identify the genes that underlie this slow clearance rate phenotype. So I'm going to tell you now about our mapping strategy, but first here's a little cartoon just about what happens in a traditional sort of hitchhiking model. So suppose we have a population of genetically variable parasites in different colors. If we have a mutation that confers drug resistance, under drug pressure, that's going to spread. And it's not only the resistance mutation that will spread, it's the whole of the chromosome if we have no recombination. And in this case, we do have recombination, so we expect to see localized reduction in variation around the resistance allele. So there's a number of consequences. As these alleles spread through populations, they carry with them linked markers, so we expect to see extended haplotypes around these selected sites. We expect to see this reduction in diversity. And also, if we have resistance spreading in one population but not the neighboring population, we are going to get dramatic divergence in allele frequencies. And they are all signatures that we can use to locate where selection has been acting on these parasite populations. So the design we've then used is to look at geographical variation. We've taken 30 samples from Thailand, 30 from Cambodia, and 30 from Laos. And we've chosen them specifically because they are neighboring populations with very different clearance rate phenotypes. And we've used a sort of two-phase test. So first we genotype genome-wide and essentially use population genetic approaches to try and locate those genome regions under selection. We're fairly confident they are going to contain these clearance rate associated alleles. So we're then just going to take genetic markers within these selected regions, genotype those in our large sample from Thailand, and we think that will be a useful approach to locate the genes involved. And this two-phase approach is quite nice in that we minimize the number of statistical tests that we need to do relative to a sort of brute force genome-wide association. So we have used a microarray-based approach to genotype. We use this because we have DNA that contains a great deal of human DNA. If we put it on an Illumina sequence, we mainly get human sequence. So this is a Nimblegen microarray with 2.1 million probes, and it assays both SNP variants and tiling probes analyze copy number across the genome. We can use just 250 nanograms of DNA with 95% human contamination. And it actually works. So when we looked at our Thai and Cambodian samples, we have 40,000 probes meet our quality thresholds, 22,000 are polymorphic, and we have almost 9,000 SNPs with a minimum allele frequency of 5% called in 80% or more of our samples. And we have 96% concordance with parallel Illumina data from 15 samples. And it detects copy number very effectively. Here's copy number at GTP cyclohydrolase, which is a well-known variant. So now we've got this data from 3 populations. We want to know where is selection occurring. And we've used 2 statistics. We've simply used FST to look for genome regions that show excessive differentiation. And we've used XPEHH, developed just down the road here, which measures essentially population differences in haplotype structure. And this was an analysis by Ian Cheeseman. So very encouraging. When we actually conduct this analysis and we identify genome regions that fall in the top 1% of statistics for both FST and XPEHH. So we then narrow down just to 2.5% of the whole genome. But within that 2.5%, we detect 4 out of 5 of the known drug resistance genes in malaria. So this really gives us an idea that we have a high true positive rate using this kind of filter. And you can see the differences, we see dramatic FST and we see wild deviations in haplotype structure. So what else do we see? We see 33 regions that fall in the top 1% for both stats. And we see a number of really strong signals. On chromosome 6, we see a strong signal. On chromosome 13, we see the strongest signal in the whole of the genome. And likewise in chromosome 14. Well, you can sit down with a glass of wine and look at the candidates in all of these regions, and a lot of them look really interesting. Their cell cycle, their ubiquination, there are a lot of really likely kind of candidates. But we think the better way now is to genotype these particular regions in a large sample of parasites with clearance rate data to actually see which one of these regions really is underpinning slow clearance. So I've told you about the samples we have, 1,733. And once we've removed multiple infections, identical genotypes in different patients, we end up with 738 samples for this genome screen. And that's ongoing, and I hope we'll have some results for Christmas. Thank you very much. So there are my conclusions. So essentially, resistance is emerging widely in Southeast Asia. It's highly heritable. Genomic comparisons can be very powerful, and targeted association of selected regions can be a very statistically efficient way to locate resistance genes.